Myself, Professor Khemchand Surana. In previous sessions, we have started with the antimalarial agents. In that, we have seen the introduction of antimalarial agent, life cycle of plasmodium species, or the life cycle of antimalarial and whatever the life cycle of malaria. Then we have seen the classification of the different antimalarial agents. Then we have started with the details of every class of these antimalarial agent. In that we have seen 4 amino quinolone derivatives. Further we have seen 8 amino quinoline derivatives. Then next class from this antimalarial agent is Cinchona Alkloids we have seen. Then further we have go with these acridine derivatives. Then Anti-folate Biguanides we have seen. Then further we have seen the Diamino Pyrimidines and sulfonamide and sulphone derivatives. So these all classes we have seen with their structure activity relationship, mode of action and their side effects. So now in today's session we will discuss about the further classes of these antimalarial agents. In that the miscellaneous class, that is what 8th class of this antimalarial agent. So this miscellaneous class is also called as quinolone methanol derivatives. Okay, quinolone methanol derivatives. So in that basically two examples, halofantrine and mefloquine. Okay, so halofantrine basically in both structure there is what hydroxyl group will be there. Okay, substitution, that's why the quinolone methanol derivatives they were called. So in mefloquine there is what quinoline ring will be there, and in case of halofantrine there is what hydroxyl group will be there. That's why they were called quinolone methanol derivatives, or miscellaneous class we can call. Okay, so the first structure, that is what halofantrine. So the basic structure is what phenanthrene ring is there. You can see phenanthrene ring with CF3 substitution, dichloro substitution, CHOH will be there. Okay, then CH2 CH2N C4H9 2 times. Okay, so this is what halofantrine structure. Next structure is mefloquine. Basic ring is quinoline with CF3. Okay, at second position, and at 8th position there is CF3. Then pyridine ring will be there with OH. Okay, CH2OH. So this is what the structure of mefloquine. Okay, so just draw the both structure and practice them. So now next the metabolism of these basic quinolone methanol derivatives or miscellaneous class. In that first the metabolism of mefloquine is given over here. So the drug which is active against chloroquine resistance strains of the plasmodium, yet cross resistance is uncommon. Okay, whatever this mefloquine will be there basically, so it is active against what chloroquine resistance strain of the plasmodium, means such strains they shows the resistance towards what chloroquine antimalarial drug. In that case these kinds of mefloquine has been utilized or the given for the treatment of malaria. Okay, but the some kind of cross resistance is uncommon. Okay, in case of this mefloquine. So this mefloquine were metabolites into what basically whatever the acid derivative carboxymefloquine derivative. Okay, so the metabolism is cited as the possible mechanism of resistance. Mefloquine is slowly metabolized through the CYP3A4 oxidation. Okay, so it is what metabolized by CYP3A4 oxidation pathway to its major inactive metabolite. Okay, so inactive metabolite is what carboxymefloquine. You can see into the structure there is what carboxylation will occur. Mefloquine is converted into carboxymefloquine. The most of the parent drugs they were excreted as an unchanged form. Okay, through the urine. So the major metabolite which is inactive, which is what carboxymefloquine. Its co-administration with CYP3A4 inhibitor means ketoconazole. Okay, if we will give this drug with ketoconazole, it will increases the area under curve for the mefloquine by inhibiting the metabolism of carboxymefloquine. Okay, so the co-administration will increase the AUC, means area under curve of this mefloquine. Okay, so how it will increase? By inhibiting the metabolism of carboxymefloquine. Now the next is what? Metabolism of halofantrine. So halofantrine will metabolize into dis-butyl halofantrine. Okay, so NH group has been formed. You can see the dis-butyl halofantrine structure. Okay, so halofantrine will metabolize into this butyl halofantrine. So halofantrine is 9 member of the 9-phenanthrene methanol class. Okay, so it is what? Phenanthrene basic structure with methanol, 9-phenanthrene methanol class, originally come out of the synthesis program dating to the World War II, but his particular agent was not fully developed until 1960s. Okay, so originally this drug is synthesized for what? During the World War II, but till 1960s it is not fully developed molecule. Further, the halofantrine has one chiral center. Okay, so in halofantrine the one chiral center will be there and has separated into its enantiomers. Okay, means they it has enantiomers, it has a one chiral center. So there appears to be the little difference between the enantiomers. This drug is used as a racemic mixture. Okay, so although it has a racemic although it has a enantiomers, but it is used in the form of racemic mixture. So halofantrine is considered to be the alternative drug for the treatment of both chloroquine sensitive as well as chloroquine resistance plasmodium falciparum malaria. Okay, so this halofantrine is again useful against the treatment of both kinds, that is what? Chloroquine sensitive as well as chloroquine resistance plasmodium falciparum malaria. Okay, so this is what their use. So it is what generally believed that the halofantrine act through the mechanism which is similar to that of 4-amino Quinolone. Okay, means whatever the mode of action of this halofantrine has been similar as like your 4-amino Quinolone. So 4-amino Quinolone mode of action is what? Basically whatever the concentration of heme, okay, means heme will converted into hemazoin. Okay, heme is toxic to the plasmodium species, but hemazoin is not toxic to the these particular parasites. So they will inhibit this heme polymerase enzyme and they will stop the polymerization of heme into hemazoin and block the conversion of these. So that is why the concentration of this heme will be increases and they will causes the toxicity to these parasites. The drug is metabolized via the N-dealkylation to dis-butyl halofantrine. We have already seen the structure of dis-butyl halofantrine, okay, here given over here. So N-dealkylation will occur and dis-butyl halofantrine will causes by CYP3A4. The metabolite appears to be several folds more active than the administration drug. Okay, so metabolites again they will appear into the several folds, okay, than their administered drugs, means number of metabolites has been formed. Okay, so this is what the metabolism of halofantrine. Now the next and last class from this antimalarial agent is Sesqueterpenes Lactones. Okay, so in that artemether and artemisonate. Okay, artemether, artether and artemisonate, these are what? The three structures from these Sesqueterpenes Lactones. Okay, the basic structure of this artemether is given over here, you can see. Okay, dioxane kind of a ring is over here. So at R, OR will be there. So if R is equal to CH3, it is what? Artemether, and if R is equal to C2H5, it is what? Artether. So the basic structure just draw and practice it. Okay, and just remember this CH3 and C2H5. If CH3 will be there, there is what? Artemether, and if C2H5 will be there, there is what? Artether. And next drug is what? Artemisonate. There is what? At OR there is OCO CH2 CH2 COO minus Na, means this is what? Artemisonate is what? Salt form. So these are what? The three basic structures of these Sesqueterpenes Lactones. These basic Sesqueterpenes Lactones, they were the artemisinin series are the newest of the antimalarial drug. So this Sesqueterpenes Lactone is from what? artemisinin series. Okay. So, and these are what newer anti-malarial agents, and they are structurally unique to the previously and currently used drugs. So, whatever the previously or currently used anti-malarial will be there. So, by structurally they were unique. The parent compound, that is artemisinin, is what natural product. From artemisinin, these sesquiterpene lactone means artemether, artemisonate was developed. So, the parent compound is what artemisinin. So, which is what natural product and which is extracted from the dry leaves of Artemisia annua. Okay. So, they were developed from what Artemisia annua, which is what sweet wormwood. Okay. So, this is what the dry leaves of Artemisia annua. From that these artemisinin was extracted, and from this artemisinin these artemisonate and artemether, these drugs were developed. So, all of the compound gives in a figure or already we have seen this compound given over here, okay, are active against the plasmodium genera that causes the malaria. Okay. So, these what active against what plasmodium species. The key structure and characteristics appears to be trioxane ring. Already we have discussed here, you can see trioxane, 3 oxygen given over here into the structure. So, the trioxane ring which consisting of indoperoxide dioxazepine oxygen. Okay. So, indoperoxide dioxazepine oxygen will be there. Note that the stereochemistry at position 12 is not chiral. Okay. So, at 12 position there is no chiral center will be there. So, these are what artemisinin derivatives. Okay. Already we have seen artemisonate, artemether, artether and artiolane. Okay. So, these are 4 what artemisinin derivatives. So, this is what about the sesquiterpene lactones. So, now the mode of action of these sesquiterpene lactone is this compound basically indoperoxide bridge. Okay. You can see the dioxane. Okay. 2 oxygen single bond, oxygen will be there, means indoperoxide bridge. Okay. Between this oxygen ring there is what between 2 oxygen there is what one bridge called as a indoperoxide bridge. So, indoperoxide bridge interacts with the heme in parasites. Okay. So, already we have seen heme is what toxic to these parasites. So, this indoperoxide bridge will interact with the heme in case of the parasites, and heme ion cleaves the indoperoxide bridge. Okay. Whatever this heme ion will what cleave this bridge, and there is what generation of reactive free radicals which damages the parasite's membrane by covalently binding to the membrane protein. Okay. Once this bridge has been break, whatever the indoperoxide bridge has been break through these heme ion, they will produces what reactive oxygen radicals or reactive free radicals. Okay. And this will what they will causes, these radicals will causes damage to the membrane of these parasites and the membrane protein. Okay. So, this is what the mode of action by structurally given over here. You can see artemisinin basic structure. Okay. Free or heme bound protein will be there. Okay. So, through this whatever the indoperoxide bridge has been break and free radical has been formed, which is what electrophilic intermediate, and further this will interacted with the particular protein of the parasite and causes the destruction or damage into the parasite. So, this is what the mode of action of sesquiterpene lactones. So, now next is what further they have given, they act rapidly. Okay. So, these sesquiterpene lactones were what act rapidly, killing blood stage of the plasmodium species and reducing the parasitic biomass. Okay. So, they were rapidly act killing the blood stage of all plasmodium species, and whatever the parasite biomass they were decreases. Artemisinin have the fastest parasitic clearance time of any antimalarial. Whatever the 8 classes we have seen, these antimalarial agents, in that these artemisinin series having a fastest parasitic clearance time. Okay. So, this point has been asked in GPAT examination. So, artemisinin are active against gametocytes, the parasitic form that is infectious to the mosquito. Okay. So they were what, infectious to the mosquito, and they their use has been associated with reduced malaria transmission. Okay. So these drug basically safe and 10 to 100 times potent as compared to other anti-malarial. Okay. So this point is also important as per your GPAD examination. Okay. So this is what, 10 to 100 times more potent than the other antimalarial agents. So this is what about the Sesqueterpene lactone. So these are what, all 8 classes of antimalarial agent that we have seen. Now from this antimalarial agent there are 2 syntheses in your syllabus. So first is what, the synthesis of amodiaquine. Okay. So this synthesis of amodiaquine is important as per your examination for 3 marks. So it will synthesize through this pathway. Okay. So the paracetamol is what, starting material. You can see the structure of paracetamol. So with this paracetamol, in the presence of diethylamine and formaldehyde. Okay. So this paracetamol were carried out the manic reaction with this formaldehyde and this diethylamine. Okay. So it will form the particular intermediate. Okay. Whatever the manic reaction is occur. Okay. At second position, then further the hydroxylation will be occur. So by the hydroxylation, whatever these NHCO CH3 bond has been cleaved and amine will be free. Okay. So further these intermediate 4,7-dichloroquinoline. Okay. You can see the structure of 4,7-dichloroquinoline. So the condensation will be occur in the presence of HCl, and the condensation between this chlorine bond and NH2 means by removal of HCl will be there, H from NH2 and Cl from this dichloroquinoline ring. HCl will be removed and condensation will be occur, and we will get the amodiaquine hydrochloride. So this is what the simple synthesis of amodiaquine from paracetamol. The manic reaction will be carried out with formaldehyde, and further the hydrolysis, and further next step is what, condensation with dichloroquinoline ring will get the amodiaquine hydrochloride. So this is what the simple synthesis of amodiaquine. Just draw and practice it. Now second synthesis from this antimalarial agent is proguanil. Again this synthesis will ask in 10 marks question, like explain the classification, SAR and mode of action of antimalarial agent with synthesis of proguanil, or simply it will ask for 5 marks or 3 marks. Because this synthesis is quite bigger, so they were asked for 5 marks. Okay. So in that again 3 steps will be there in the synthesis of proguanil. So the first step is synthesis of parachlorophenyl guanidine. Okay. The first step is what, synthesis of parachlorophenyl guanidine. So the starting material is, you can see, 1-chloro-4-nitrobenzene. So in 1-chloro-4-nitrobenzene there will be all reduction will be occur. The nitro group will convert it into amino, means 4-chloroaniline. Further these 4-chloroaniline will treated with CNBr. Okay, cyanobromide. Okay, you will get 4-chlorophenyl cyanamide. Okay, so NH2 will be converted into NHCN. Okay. Further this will treated with ammonia in the presence of hydrogen. You will get parachloroguanidine. So this is what, first intermediate, parachloroguanidine. Initially with this nitrobenzene reduction will be there, converted into 4-chloroaniline. Further with ammonia treated, you will get parachlorophenyl guanidine. So this is what, first step, synthesis of parachlorophenyl guanidine. Now second step is what, synthesis of isopropyl cyanamide. Okay. So isopropyl cyanamide. So for this the starting material is what, isobutyric acid. So this isobutyric acid CH3CH3COOH will treated with this SOCl2, whatever the acid will convert it into acid chloride. Okay, COOH will convert it into COCl. Further this will treated with ammonia NH3. whatever COCl will convert it into amide. CONH2 means acid chloride will convert it into amide. Okay, from acid it is converted into amide through acid chloride. Further, this amide, isobutyric amide, will decarbonylation reaction will occur and you will get isopropyl amine, means amide will convert it into amide. Again it will treated with this cyanobromide CnBr, it will convert it into isopropyl cyanamide. So this is what second intermediate, this step is what synthesis of isopropyl cyanamide. So step first, now step second. Now next step 3 of the synthesis of this proguanil. So we have synthesized the intermediate 1 and intermediate 2 from step 1 and step 2. So step 3 is what? Condensation of product of step 1 and step 2. So the product of the step 1 is para-chlorophenyl guanidine and the product from the step 2 is isopropyl cyanamide. So both these product will be condensation undergo under the condensation reaction and there is what? Formation of proguanil. You can see the structure of proguanil. Further, this proguanil will treated with hydrochloric acid, means HCl, it will convert it into proguanil hydrochloride. So this is what the synthesis of proguanil hydrochloride. So these are what? Two syntheses. The first is what? Synthesis of amodiaquine we have seen, and second is what? Synthesis of proguanil. So these both synthesis is important as per your examination. So now here this chapter has been over. So thank you. In next session we will start the new chapter. Till that, happy learning.